Catherine, after class, remind me to mark you not absent, okay? So, we talked about the lac operon, and I'm pretty sure we talked about this part of the control of the lac operon last time we were together. So let me just give you a little bit of quiz and see what you can tell me about the lac operon. If the promoter region looks like this, is transcription going to happen? No, why not? The repressor's blocking, right? All right, good deal. So transcription can't happen because the repressor is blocking. What has to come into the system to kick the repressor off in the case of the lac operon? The allolactose, and we actually call it an inducer, right? It comes and binds the repressor, makes the repressor change shapes. It can't hold on to that operator sequence anymore, so it falls off. So then the next question is, if the promoter looks like this, will transcription happen? Will it? Can't. So that's why I'm giving this quiz, to see if I talked about this fully enough. If the promoter looks like what's circled there, transcription still will not happen. That's what the promoter looks like when there's lots of glucose present in the environment. Because remember, when there's lots of glucose present, there's one other molecule that's going to be very low abundance in the cell. Cyclic AMP. If there's lots of glucose, cyclic AMP levels will be very low. And remember, at this particular promoter, you need one more protein to come and bind here. That's that cap activator protein. It has to bind, but it will only bind if the cyclic AMP level is high enough. It can't bind if there's no cyclic AMP. And so when there's glucose in the environment, there will be very, very little cyclic AMP, so the cap activator protein is not able to bind onto that region, and it can't transcribe. So basically, that cap activator protein is necessary to come in and stabilize RNA polymerase on this promoter. RNA polymerase can't bind to this promoter and can't start transcription unless the cap activator protein has come and bound here, and it can only come bind if the cyclic AMP level is high enough in the cell. Once this scenario happens that I just depicted there, once all of this happens, then transcription will begin, and you'll start making transcripts, and RNA polymerase will run down and continue to make transcripts until what would happen? What might change it? What might shut it off? Well, the transcription is going to stop down here at some transcription termination, but what's going to shut off this promoter region and make it stop making those transcripts? One would possibly be if you get more glucose in the environment. If you do that, there'll be no more cyclic AMP, so it would stop that way. But let's assume that there's no glucose in the environment. What will happen to make it stop in that case? Remember, all of these control mechanisms have to be turned on, be able to be turned on, and have to be able to be turned off. So what would shut off this gene? What would make it stop? One is glucose coming into the environment. That's a possibility. What's the other possibility? Absence of lactose. So if there's no lactose, there's not going to be any of that allolactose, and then pretty quickly you're going to go back to this top state where the transcription turned off again. Okay, beautiful. I think you got it. So now we can talk about the next slide that I had started on and make sure that we're there and we're on that together, because the next slide has this beautiful growth chart, graph of growth, and it looks a little bit like what you've seen before. You see the lag phase, and then you see a log phase, and then previously we saw a stationary phase, and then death phase, and then prolonged decline, something like that when we looked at this type of growth phase. But now we're seeing two different, apparently close to logarithmic phases. And so this type of growth curve is called a dioxic growth curve. In other words, there's two different carbohydrate sources in the media. One is glucose, like we always consider, and the other in this case is lactose. And you just learned from talking through that last figure that if there's glucose present, the cell won't use the lactose. It won't touch it at all. Remind me why that is. If there's glucose present, why won't it use the lactose? It prefers glucose, and what's going on at the lac operon that will not allow it to to use the lactose if there's glucose present? There's low cyclic AMP, so the activator protein can't bind to that to the activator binding site, and so transcription can't start at the lac operon. Okay, good deal. So now we can think about this growth curve. I'm going to do it again. Sorry. Starting to get a little cold. So here we've got this situation. Let's think about each one of these phases of this growth curve. What's going on right here? Here we're going to think about what's going on at the promoter region of the lac operon. So we're thinking again, is the repressor bound? Is it not bound? Is the cap activator protein bound? Is it not bound? So in that first phase, that first lag phase, what's actually bound there at the lactose promoter? Is the repressor bound? Is the repressor protein bound? And then we also need to think, is transcription happening? So is the repressor bound in this case? We start out, and this media has both lactose and glucose in it. And that's what's in the media to start with. So is the repressor going to be bound if there's both lactose and glucose present? I heard a yes and I heard a no. If it's yes, why would it be bound? If it's no, why would it be bound?
Because there's no lactose being produced and there's no cyclic AMP either.
How do we get allolactose produced? It happens spontaneously whenever there's lactose in the environment. Some of it gets converted to allolactose. So in this first case that I've got circled, would there be allolactose?
Yes.
There should be allolactose, right? Because there is lactose. And so would the repressor be bound?
No.
The repressor would not be bound because there's allolactose. It's going to kick the repressor protein off of that operator sequence. What about the cap activator protein? Will it be bound?
No.
Why will it not be bound?
Because it's cyclic AMP being used.
Cyclic AMP should be low because all of the ADP will pretty quickly be recharged to ATP. None of it will get all the way discharged to cyclic AMP. All right, good deal. So that's what's going on in that first case. And so the third question is transcription happening in that first phase, in that first log phase? At the lac operon, is transcription happening? No, it can't happen because the cap activator protein's not there. So here we got no transcription happening at the lac operon. Okay? What about this next phase right here? This first exponential phase. Still glucose present, still lactose present. Is there any transcription happening at the lac operon? No transcription at the lac operon. Why not? Still plenty of glucose, so all the ADP is getting recharged to ATP, very little cyclic AMP, and there's still lactose present, so allolactose is present. Okay? So at this point, from that point on, right there, those first two phases that we talked about, remind me again, is the repressor protein bound to the lac operon or is it unbound? It's unbound, and again, why is it unbound? Because there is allolactose present, because lactose is present. Whenever you have lactose present, some of it's going to get converted to that similar molecule that's called allolactose. If you want to know what it looks like, go to Wikipedia. They'll show you a beautiful picture of what it looks like, and you can look at lactose and see how it looks. Not too much difference between those two molecules. All right, then we hit this third phase right here that kind of looks like that stationary phase that we saw previously. And at that point, the glucose at the beginning of that phase, the reason it starts is because the glucose is used up. The glucose is exhausted. So if we go back and look at the lac operon again at the promoter of that, is the repressor protein going to be bound at that point?
No.
Why won't it be bound?
Lactose.
Because there's lactose present, so there's going to be allolactose. That's going to kick the repressor protein off. Will the cap activator protein be bound?
Yes.
Should start to be bound, right? Why should it start to be bound? Because the glucose is used up. That means cyclic AMP is going to start to be produced, and when you get enough of it, then the cap activator protein will be able to bind, and then transcription can happen. So that's exactly what's going on. That's why you get this lag phase. This lag phase, the cells are sitting there, and they're sensing the system, and they've got to kick on the operon. They've got to make that messenger RNA. They've got to translate those proteins, and the proteins have to get to the places they need to get to in the cell so that the cell can now use the lactose. And so talk about what's going on there. And now we've got this second seemingly exponential phase. You'll notice that the slope on that second phase is actually a little bit less steep. And that's because the cells grow less well on lactose. They can get less energy out of it, therefore they can't grow as fast. And so you get a phase that's actually a little bit less steep than the first exponential phase. So in the middle of this phase, they're growing on lactose. What do you think about the lac operon? Is the repressor bound to the promoter region, the operator region in the lac operon? No. Okay. Is the activator protein bound to the promoter region? Should be, right? Why? Cyclic AMP levels are still high. They're not getting as much energy out of the lactose as they could out of the glucose. The cyclic AMP levels are still relatively high. And so are you having transcription from, you are getting transcripts made from the lac operon. Beautiful. So that's that phase. And then we get up here to this point, and now all the lactose is used up. Now there's no glucose, there's no lactose. Let's look back and think about the lac operon at that point. All right, so the lactose is used up, the glucose is used up. Is the cap activator protein going to be bound at the activator binding sequences? I heard a no. Does everybody agree? What makes it bind? The level of cyclic AMP. No glucose, no lactose. What's going to happen to the level of cyclic AMP? It's going to go up even higher because now there's no carbohydrate to be used to convert ADP to ATP, and all the ADP is going to get kicked down into cyclic AMP. So the cyclic AMP level is going to go up even higher. So in that case, right here, the cap activator protein is still bound at the lac operon, at the activator binding site. Right? Still bound there. It wants to transcribe if it can, but what else is bound there at that point? Now there's no lactose present, so what else comes and binds? So the repressor protein comes and binds at that point. So even though the activator protein is bound in this scenario right here where there's no glucose and no lactose, transcription cannot happen because transcription is blocked by that activator, or by the repressor protein that's bound there. So let's think about this. You're a nice student. You hate to see bacteria suffer, and you see them suffering right up here at the top. There's no carbohydrate, and you decide to throw some lactose into their environment, and so you give them a nice dose of lactose so that they can start to grow again. What would happen as soon as you put the lactose in, based on what you just told me about that lac operon promoter right now? You said the activator protein is bound, the repressor protein is bound. So you throw a little bit more lactose in, what's going to happen? Beautiful. Some of the lactose will be converted to allolactose, and then the allolactose will deal with the repressor and cause it to change shapes, and then it will drop off, and then transcription will happen again, and then the cell will be able to use the lactose. Let's switch gears and think about it from a different perspective. You're still right here. You throw in a little glucose because you're really nice to these guys. What's going to happen then? They're obviously going to use the glucose, but what's the lac operon? What's the promoter going to look like in that case? Will the repressor protein be bound if you throw in glucose? Yes. Why? Because there's no allolactose. You threw glucose in. There's no lactose, so there's not going to be any allolactose to kick the repressor protein off. Beautiful. What about the cap activator site? What will happen there? It won't be bound either because the cap activator protein won't bind. Why won't it bind? Because pretty soon the cyclic AMP level is going to go down, and then they'll cut off transcription at the lactose operon once the cyclic AMP level goes down.
So what takes the cyclic AMP from the site?
So every chemical has an association constant and a dissociation constant. And so when, just like what we talked about with antibiotics, if you take them and keep the levels high in your bloodstream, then there's plenty to fill those enzymes' active sites and block them by that competitive inhibition we talked about. In this case, if there's enough of the cyclic AMP, it'll associate, but when it drops off, all the free cyclic AMP drops off, pretty soon that one that's bound to the cap protein is going to dissociate. There's no more to come and reassociate with it, then that protein's no longer active until another molecule comes and binds to it. So it's that same kind of scenario. Good deal. You guys got it. I think you should do well on the exam on that part.
So with this, transcription still happening, just not by the
Sure. We're just focusing on this one operon, but of course all those enzymes that are needed for glycolysis are still being transcribed, and many other genes are also being transcribed. But we were just focusing down on this one particular gene set that we call the lac operon when we were thinking about those scenarios. So yeah, certainly transcription's going to happen. If this was Bacillus cereus and it got to this point, then a lot of genes would kick on that normally were repressed. What kind of genes do you think might kick on? Yeah, the sporogenesis genes would kick on at that point because they're thinking, uh-oh, we're in trouble. There's no more carbohydrates here. This is not going to be good for us. We better save ourselves. And so they'll kick on all those normally repressed genes that are sporogenesis genes. And then they'll start doing transcription and translation from those genes to be able to make the proteins that are needed to go into sporogenesis. Good deal. All right, so this is just a little bit more of the story of the lac operon. There's a protein that brings glucose into the cell. We actually talked about this scenario way back in chapter 2 when we were talking about how molecules get into the cell, and we talked about one of those methods where the chemical that's outside is brought into the cell, but as soon as it gets in the cell, it gets changed. And in this case, we see glucose getting changed and being phosphorylated to glucose 6-phosphate, which is that starting point that we talked about at the top of glycolysis. That's the first step of glycolysis. So this helps with glucose coming into the cell because as soon as it's brought in, it's no longer glucose, and now there's still lots and lots and lots of glucose on the inside and no glucose, on the outside, and no glucose on the inside of the cell. And so that keeps that concentration gradient of glucose high on the outside and little on the inside, and it helps the process of moving the glucose across to the inside of the cell. There are specific permeases that help to do that, but having that concentration gradient is definitely a pressure to bring the glucose in toward the cell. Okay, so they get concentrated inside. When we shift over to this scenario, if there's less, then we begin to see some other things happening down inside of the cell. And this is what we just talked about with that cap activator protein right here, and what's going on with ATP and cyclic AMP over here. If cyclic AMP is high, then you'll begin to get the activator protein bound by cyclic AMP. It'll jump over here and it'll bind over here, and then transcription will happen on this gene. But again, it can go backward, and if cyclic AMP becomes low, then that whole system is not going to work, and you won't get any transcription happening there because this cap protein won't bind there. Okay. All right, good deal. Just realized the lights are pretty bright. Let me see if I can fix that.
So when we went over gene regulation, we went over inducible and repressible.
Great question. I love that question. What do you think? Is this gene inducible, or is it repressible, or is it both?
Seems like both, but if I had to choose, I'd say repressible.
So if you're growing the organism under normal conditions like it would experience in the environment and there's glucose present, is this gene turned on or off? It's repressed. So it's not normally expressed. And so it's not normally expressed because the repressor protein is going to come and it's going to bind right here. And it's going to block that RNA polymerase. And that's the normal situation. Because in normal life for bacteria like E. coli, there's not a lot of lactose present. It's not a prevalent carbohydrate out in the normal environment. So normally this gene would be repressed. But then you ask another question, is it inducible? And yes, it has to be induced to be able to be turned on. And the induction part is right there. The induction part is this guy. It's only induced if cyclic AMP levels get high down inside of the cell. So the answer to that question is actually both. It's repressed normally, but it also has to be induced to be turned on. Removing the repression isn't enough to turn it on. You also have to induce it. And that's basically what we talked about when we walked through this last figure right here. We were talking about which states were in repression and which states were being induced. So, great question. That kind of ties everything back together. What was that?
Do they normally go together?
It just depends on the particular gene, the particular promoter, and what's going on in that situation. But the reason many textbooks use the lac operon is because it does have both. It requires both. And so you can get the concepts of how they can both work together. Some genes are usually just repressed. You kick the repressor protein off and then transcription happens. In that case, what would the promoter have to look like? If all you have to do is kick the repressor protein off of that promoter sequence, what would be different about that promoter than, let's say, this promoter? Check out this promoter. This promoter requires this interaction. It requires the cap protein to bind there. Why does it require it to bind there? back a couple slides, but remember I told you if the cap protein is not there, RNA polymerase can't bind and it'll just fall off. It won't associate strongly and transcription won't happen. That's what happens when glucose is high and lactose is high. In that first scenario we talked about, transcription doesn't happen because RNA polymerase can't bind tightly to that particular promoter. And remember, promoters are just a certain combination of A's, T's, G's, and C's, and sometimes they look like that, but sometimes the sequence might look like this. Whoops, that wasn't a C that I wrote the first time. And sometimes they might look like this, or sometimes and there's about 70 nucleotides that make up a typical promoter, and sometimes one nucleotide change from one that works constitutively, if we say this first one is constitutive, changing that one nucleotide might make the promoter still work, but it'll work just a little bit differently. And the RNA polymerase that comes in might not bind to this guy very efficiently, and so it might need some help. It might need the help of some other activator protein to come in and bind, like we saw in this scenario here on the bottom right, where we need this activator protein to come in and bind and help the RNA polymerase stabilize there so transcription can happen.
So it has more of an affinity to the cap?
Beautiful. Remember enzymes? Enzymes typically have one substrate, and RNA polymerase is an enzyme, and it has to bind to a substrate, if you will. Usually we think substrates get changed in the process of a chemical reaction, but it's got to bind to that promoter sequence. And there's hundreds of different possible promoter sequences. Some of them bind RNA polymerase really strongly. Others of them bind RNA polymerase really weakly. And this one needs the help of this cap protein to help it bind strongly enough that it can get there and start the transcription process. So the cap protein has to be there. Remember we talked about sigma factors and how sigma factors help the RNA polymerase find certain promoters. Sigma factors are doing kind of the same thing, except for they usually interact with the RNA polymerase, change its conformation just a little bit so it can find a certain promoter or a different promoter. And this guy is specific for this one. It binds there and says, RNA polymerase, come on over here. This is a good place to be. Set down here. Hopefully you can do some transcription here. And it recruits the RNA polymerase over to that part of the chromosome. In bacteria, those activator binding sites tend to be right next to the promoter. But in your chromosomes, they can be thousands. Some of them are 2,000 base pairs away. Some of them are 10,000 base pairs away. And usually they are upstream. I think I told you this last time. Since RNA polymerase moves this way from left to right, the way we usually diagram a gene, we call this area right here downstream. And we'll call this area up here and up here upstream. So things that are over on this side we'll call upstream of the promoter, and things that are over here we'll call downstream. And most activator binding sites are going to be upstream. There's a few that are down, but most of them are going to be up somewhere in front. And that somewhere in front might be very close, like in bacteria, or very far away, like in eukaryotes. Those proteins still bind to that area, and sometimes it's multiple proteins. Part of my PhD thesis was looking at one organism and one gene set and trying to figure out what the proteins were that bound to the sequence upstream and how they interacted with that promoter and got RNA polymerase to come to that promoter and do the transcription process. So, interesting parts of regulation. If you think about your chromosomes, you don't have operons on your chromosomes. You've got genes scattered sometimes on multiple chromosomes, but they have to work like operons. In other words, they have to all be turned on at the same time. So on those organisms, those higher organisms, eukaryotes like us that have multiple chromosomes, how does the cell know, turn on this gene on chromosome 1 right now and do the same thing for this gene on chromosome 5 and this one on chromosome 15? And it's a really complex picture, but they've used very similar systems to do that same process. And in that case, the promoters for each one of those genes that needs to be turned on at the same time will look very much the same. They'll have the same promoter sequence, they'll have the same or similar activator sequence, and they'll have some other characteristic things that are similar between them. And so that way our cells can regulate that process of gene expression. And one of the other things that regulates expression that we're finding out in the last 5-10 years is what's going on with chromosome structure. We call it chromatin structure. How is the gene packed? And there's a number of things that control gene expression. We didn't know it until about 5 years ago, but those nucleotides, we knew they could be modified. They oftentimes get methyl groups put on them, and depending on how they're methylated, describes and dictates how they can be transcribed. So more methylation tends to result in a different level of transcription than others, and that's what this is talking about, modification of chromosome structure. Then there's all kinds of regulation of transcription. We just talked about how that can work, and in the eukaryotes I just told you it's much more complex. And then we also get this situation where we get a pre-messenger RNA that's produced, and then processing that, remember that's capping, that's polyadenylation on the 3' end, and that's splicing. Those things happen, and then any post-translational modification of the protein that's made, glycosylation, clipping off that signal sequence that we talked about, and some of the other things we've already talked about, those typically have to happen in eukaryotic cells. But think about this scenario. You're a cell, you're growing along, everything's good, you're making RNA for a specific gene to make a specific protein, and now all of a sudden you don't need any more of that protein. But every time you make it, you're pumping in 4, 5, 6, 800 amino acids that sucked energy out of the central metabolic pathway. You're wasting energy if you keep making that protein. But you've got this RNA in the cell, so how do you stop that RNA from being used for translation? How do you get rid of the RNA so that now the ribosomes don't have access to it? And that's where this process that's called RNA interference, and sometimes we just abbreviate it as RNAi, that's where this comes into limiting the amount of RNA that's in the cell so that translation of that RNA will no longer take place. And RNAi, or RNA interference, is an interesting phenomenon that's really just begun to be understood in the last 5 years or so. If you go back 10 years in the literature, you won't find any discussion of it. We were starting to see some small heteroRNAs in the cell about 10 years ago, and people were wondering what's the function of these RNAs, what are they doing down in the cell. And now we know one of the functions is this process that's described as RNA interference. And RNA interference works like this. A cell's growing along, and now the cell has all of that gene product, all that protein that it needs. And it doesn't need to make it anymore. It doesn't want to waste the energy in that direction anymore. So it needs some way to shut it down, and it'll begin to produce these very small RNA strands. They're single strands. They tend to be just small strands, 10, 20, 30, 40, maybe 100, but 100's getting pretty big molecules of RNA, and those small RNAs bind to this protein complex that's called RNA-induced silencing complex, or RISC for short. RNA-induced silencing complex, and those little RNAs will be bound by this protein complex, and then this protein complex will find messenger RNAs that have sequences that are matching or homologous to that little short RNA that's being made down inside of the cell. And then this complex actually clips the RNA, and it breaks the RNA and cleaves the messenger RNA right here. And when that messenger RNA is cleaved, then it's broken into two different parts, and now the messenger RNA is no longer functional anymore. Remember, the messenger RNA was capped out on this end with that methylated G, and out here it's got polyadenylation, and the reason those were added is so that the nucleases couldn't cleave and chew up this RNA. But now it's clipped in the middle, and the nucleases can grab this end on this piece and start chewing away from this end toward the 3' end. And other nucleases can grab this 3' end of what's left here and start nibbling away nucleotide by nucleotide over here, turning them back into free nucleotides that can be used for transcription of additional messenger RNAs from some other gene. So that's the way this process works. Very specific RNAs are made that are homologous to messenger RNAs that are there. They can base pair, and then this complex clips them, breaks them into two parts. Then the messenger RNAs can be broken down.
So messenger RNAs can be read more than once?
Sure. As many times as ribosomes jump on there. I think we've already talked about in prokaryotes where transcription is happening. This is the DNA, and then if this is the RNA that's being transcribed, and so RNA polymerase is up here, and then we talked about how the ribosomes will grab onto the ribosome binding site and they'll migrate that way, and so you might have a bunch of ribosomes on there. The same thing can happen in your cells with multiple ribosomes latching on. It's just all linked together here in this prokaryotic cell, but once the RNA gets out to your rough ER or the free ribosomes, then multiple ribosomes can use that same messenger RNA. Yeah.
With the polyadenylation in the cap,
Uh-huh.
that prevents the nucleases from breaking it down, but eventually it'll break it down, so it'll keep producing until those ends get chewed off.
It'll keep producing until something happens, but this system actually cuts it right
this is different.
right here. So this is the main system for shutting off RNAs unless they somehow break, some enzyme does break them down. And this is one enzyme complex that specifically breaks them down. All the others are non-specific mechanisms that will pretty much chew up any RNA. This one's specific for one specific message because this primer has to match the RNA that's going to be destroyed. It won't destroy all the RNAs in the cell. It'll only destroy specific RNAs down inside of the cell.
So that's the only way to break up.
That's the way that we know of right now that RNAs are removed. But like every scenario, once you figure out one, it's easy to start looking for others. And there probably are some other systems, but this is the most recent one that's been observed to specifically degrade RNAs that are no longer necessary. It's a big question for a long time. We make these RNAs, they're pretty stable because they're set up to be stable, and how do you stop making the proteins from them if you don't need that protein anymore? And so they started looking, and then they figured out, okay, there is a system down inside of the cell that RNA research is pretty hard because I already told you there's lots of enzymes everywhere that chew up RNA, and so making this discovery was actually pretty complex because as soon as you start looking for things that are chopping up RNA, you don't know if it's a contaminant that you put into the system or if it's something that's really happening down inside of the cell. And so it took a lot of really high-tech science to be able to figure this out.
I don't know why I was thinking this, but you don't make a new strand of RNA every time you eat a specific protein.
Well, it depends. If we go backward to that promoter, it depends how strong that promoter is. Some might just make a few strands of RNA. Some might make hundreds of strands of RNA, again, depending on how strongly RNA polymerase binds to and how much it's recruited to that promoter sequence. So there are different levels of transcription, which is what I think I just heard your question to be. And that's very true. We already know we can shut it all the way off. We can turn it on. There are intermediate levels, and some ons are much higher than other ons. And so that was part of my PhD thesis, too, to try to figure out why one gene of interest was actually sometimes being turned on very high in the cell, and other times it was just on at a basal level during life cycle. And so, yeah, there's lots known about that as well. And, yeah, you got it. All right, so switching gears just a little bit, we need to get to genomics. We've talked about this a little bit before. Basically, genomics is the study of genomes and sequencing genomes. So in the old days, DNA sequencing was really slow, laborious, but now there are systems that are called high-throughput systems. And I think I told you it cost multi-million dollars to sequence the first human genome back in the 90s. It was going on, and the project had already started, and it cost millions and millions and millions of dollars. But today it's estimated to cost about $2,000 to sequence a human genome. And in 5 years, it's estimated that it'll cost about $500 to sequence the entire genome. So it'll probably become a standard routine medical test. Because if you sequence the genome and you know which genes tend to lead to cancer or other diseases, you can go in and look and see, does this individual have a mutation in this gene that usually leads to the progression of this particular disease? Sometimes you can even predict that this person's going to have this disease, and if we see that in advance, then we can maybe do some therapy where we can correct that mutation and fix the mutation so the individual doesn't actually progress to that disease. And the old adage that an ounce of prevention is worth a pound of cure is, I think, very true. If we can figure out a person's about to have a disease and we can fix the problem that might lead to that disease, then that's oftentimes much easier than actually fixing the disease. And so we started way back in the 80s, started sequencing genomes of the entire genomes, the entire chromosome or chromosomes of individuals. And this guy, Haemophilus influenza, is one organism that was of interest because it causes a disease called influenza. It's a bacteria that causes another type of influenza. And so this particular bacterium was sequenced completely and reported in 1995. And since then, literally thousands, probably hundreds of thousands of different entire genomes have been sequenced. I told you already, for sure thousands, because they've gone in and they've sequenced all of the genomes of all of the thousands of bacteria that live in your gut. They know in the average individual there are over 1,000 different species of bacteria that are living in your gut, even when you're healthy. They're doing stuff for you like making enzymes to break down things that you can't normally do and all kinds of things. And we really want to know why they're in there. What are they doing? How are they helping us? At this last meeting, I don't think I told you this story, there was a couple of papers that talked about, remember when we talked about hamburger and how much nitrates are being put into the hamburger and how that's probably not healthy for us? In this last conference I went to, there was one group that was presenting, they made a couple presentations. There are bacteria in your gut that actually can synthesize nitrates. There are others that can take those nitrates to nitrites, and others can take them to nitrosamines. And the nitrosamines are the ones that actually cause cancer. So if you have bacteria that are in your gut, and there's lots of them, and they're making nitrosamines, does that actually lead you toward the direction of actually having cancer? Is it the bacteria that are having cancer? One hypothesis is we're pretty nice to the bacteria that live in our gut when we're alive. We feed them regularly. But they constantly have to fight against an immune system that wants to kill them, and they keep them only in the gut, hopefully. But when you die, those bacteria no longer have to fight against the immune system. The immune system dies along with your body. And then the bacteria that are in your gut, if you're not cremated or embalmed, they've got all those nutrients that are in that rich bag of nutrients that you call you, and they have free reign over that bag of nutrients. So are they actually trying to say, hey, it'd be better for me if you just die by making some of the chemicals that they make down inside of our gut? And we want to know that. And so there's projects that are actually sequencing all of the bacterial genomes that are in the guts of literal thousands of individuals right now. And then they want to try to follow those individuals over time and say, okay, they've got these bacteria. How long was their lifespan? What disease did they succumb to see if they can get any pattern of comparison between the bacteria that are living in the gut and diseases that we progress to. And I think that personally there probably is a lot of relationship between what's going on in the gut. The bacteria, if you look at just sheer numbers, there's about 10 to 100 times more bacterial cells in your gut than all the other cells that make up your body. And they're doing all kinds of metabolism, many different kinds of metabolism than your cells can do. And they're wanting to have a nice place to live, and they're trying to build that home to be comfortable for them. And so it's easy to imagine that they are influencing what's going on biologically and possibly even what's going on genetically in your body. When we get to the next chapter, you'll figure out that genomes are not as stable as you've always heard they are. Bacteria share their genomes. One guy will die, his DNA will be shed in the environment. The next guy, maybe the same species, but very likely a different species, will go and grab that DNA, bring it in, and save part of it because it thinks it might help it along the way. And so we start to see that even species are very different. People have sequenced species or strains of E. coli, and they've sequenced tens of thousands of strains, the entire genomes, and they see that no two are alike. Each one has done something a little bit different and pulled in different bits of DNA from the environment. That's how we got that one strain, E. coli 0157H7, that's very lethal to humans. It picked up a big chunk of a chromosome from one of its cousins, brought it in, and that chunk of chromosome has special toxins that E. coli never had before until it got those toxins from a piece of DNA that one of its cousins shed into the environment. So we'll talk about that down the road, but that's what genomics does, and that sprung up a entirely different field called bioinformatics. This is probably where I would have gone if I was studying and today where you are. Because I always loved computers, but when I was an undergrad, computers were so cumbersome. The only computers were mainframes, and you had to punch these cards, and each word or each comma had to be on a separate card, and you'd punch a stack this big, and you'd give it to the people behind the desk, and they'd say, come back tomorrow, and we'll tell you if it worked or not. And you come back tomorrow, and you found out there was one typographical error called a syntax error. And so you'd fix that one, and you'd look through the rest of the 2,000 cards that are in your code, and you'd give it back to them. They'd say, good, come back tomorrow, and we'll tell you if it's good or not. And 5 or 10 cards later, you missed a typo, and it didn't work again, and so you had to fix that one, and once again, you'd look through them all, and you give it back to them, and it would just take weeks to get any simple code written that now you can do in about 10 or 15 minutes on your own PC in your home if you want to. And so bioinformatics is doing that. It's tying computer systems to analyze all this genomic DNA information so that we can start to take the genomic DNA, those chromosomes, and say, okay, we can see this chromosome and we can see what's going on. My pen's not working. Oh, I must be touching the pad. That's what's going on. So we can see this chromosome, and here there's an AUG, and here's that's a G, and that's a stop codon, and then over here there's another AUG, and look, they're going in the same direction, so maybe that's an Operon, and over here there's a stop codon here on the other strand, and there's an AUG, so that one likely gets transcribed in the opposite direction, and you can begin to look at the chromosome and map out. We call it chromosomal annotation or gene annotation. You can begin to map out where the open reading frames are on that chromosome. And then you can take the sequence of those open reading frames and you can throw it into a computer, and the computer will spit out and say, if this were used to make a protein, it'll make a protein that has this amino acid sequence. And if it's used to make a protein with that amino acid sequence, that protein looks a lot like these proteins that we know function in this way. And then you can say, okay, that must be a putative gene for RNA polymerase. It looks a lot like the other RNA polymerases that have been characterized. That's probably what it is. And the computer will tell you it also looks a little bit like this particular gene that makes this other type of molecule, and it'll give you a best guess scenario. It's either this or this, and here's the probability that it's this, here's the likelihood that it's this. And that's what this bioinformatics is, trying to use the information that we get from biological analysis, in other words, genomics, to be able to determine what organisms in the environment and in our bodies are able to do. It's a really interesting field. If you love computers and computer programming at all, lots of money is being pumped into that direction because so much money is being pumped into genomics and understanding all those microorganisms. The cool thing about genomics is that we can go to places and grab bugs that we can't grow. Those thousands of bugs that are in your stomach, we can grow about 150 of them. The rest of them are so fastidious we don't know what they need. We can't give them the environment that we need, but we can go grab DNA out. We can extract DNA, we can sequence their genomes, and even without growing them, we can figure out what they're capable of doing. You can go to the soil, you can go to any environment and look and see who's there. You don't even have to grow it. Just isolate the material, extract DNA, sequence all of the genes in the DNA. Computers glue it all back together and says this goes with this and this goes with this, and starts putting it together in circles if it's a circular molecule, or linear if it's a linear molecule. And so that's what bioinformatics is. When you're doing genomics, we did genomics way back when I was sitting where you are too. We were analyzing the chromosome, and we were specifically looking at DNA sequences. Looking at DNA sequences and looking for those sequences that might be open reading frames. So we're going to read the positive strand because it's the same sequence as the transcript that's going to be produced. Even though it's the negative strand that's the template for transcription, the positive strand has the same sequence except for A's being replaced with U's. And so in the DNA there's going to be an ATG that we're going to see, and that can be translated into an AUG, a start codon. And you begin looking for these ORFs, or open reading frames, and that's what you're looking for. A place where there's an AUG or ATG if you're looking on the chromosome, and then some stop codon down at the end. And then you've got X number of nucleotides, or X number of amino acids coded for in between those start and stop codons. That's a definition of an open reading frame. And that's what you're looking for when you're just going in and sequencing genes. Usually you look for stretches of nucleotides that have at least 300 base pairs in between the AUG and the start codon, because that's a pretty small gene. It only makes a protein of 100 amino acids, and most proteins are larger than that. So usually you're looking for something that has that open reading frame that goes longer than that. And then it's going to end with a stop codon on the other side. So you guys are inquisitive, so I know you're going to ask, why do you call it an open reading frame? And so you're looking on the DNA, and you're reading the DNA, and over here you see an ATG, and down here you see a stop codon. And the reason we call it an open reading frame is because this stretch between here and here has no other stop codons in the middle. If there's a bunch of stop codons in the middle, then that's not an open reading frame. It's blocked by those stop codons. The reading frame is not open because there are stop codons in the middle. But if those stop codons weren't there, then that whole stretch would be open to be read for translation, and then you can potentially make a protein that's the length between that start codon and the stop codon. Yep.
On eukaryotic DNA, you've got the 3' and the 5' ends, but in prokaryotic circular, as far as I know, it doesn't have that. So can prokaryotic DNA, can it be read in both directions?
So whenever we sequence DNA, usually we sequence both directions. We sequence both strands, in other words. Because there's enough error if you just sequence one that you're likely to find a mistake and not realize it unless you sequence both and align them up together. But I think your real question is, where do you start in sequencing? And there's all kinds of different methods that are out there. And so the question then, in the original system for sequencing DNA, and that's probably all I have time to tell you about, it worked this way. Remember that one characteristic of DNA polymerase? It can't start de novo. Can't start synthesizing de novo, and that's actually a fortuitously good thing, because now we can make little primers that will bind to one strand of the DNA and maybe set down right here, and this primer has a 5' end here and a 3' end here, and if we put that primer in, and if it's specific enough that it can only find one place in the chromosome to do this annealing and hydrogen bonding, then we can throw in DNA polymerase, and DNA polymerase will come in right here, and DNA polymerase will then grab right there, and then DNA polymerase will come along and once it binds, start synthesizing the rest of the nucleotides, and it's synthesizing in this direction, 5' to 3'. And we've got systems to stop various strands that are synthesizing at different places, and then we can figure out the sequence from that. There's lots of different scenarios that play along differently. Some of them now don't even need this primer. They do things a little bit differently, and then you can start sequencing all along the chromosome, and the computer figures out what's going on with each one of the strands that are happening. The technology has advanced quite a lot. So, like I said, the first time we sequenced the human genome, it took literally close to 10 years. Now it can be done in about 2 days because of the advances in technology and the speed at which the sequencing process can happen. So again, the technology, and pretty soon, I imagine in one day they can swab cells out of your cheek, take those cells in the morning, and by the end of the day they'll have your entire genome sequence from those cells that are in your cheek. It's just advancing that quickly. And again, they'll be looking for these open reading frames, and then we know which open reading frames are important for cancer progression. They'll be looking at all of them, and the computer will spit out and say, uh-oh, there's a mutation in this open reading frame, and when you get a mutation in this open reading frame, this guy might get lymphoma. Or if you get a mutation in this one, they might get some other disease, and then hopefully by then we'll have ways to correct those mutations and be able to fix that progression. Yeah?
I think eventually they'll use that at childbirth. They just look at your...
Yeah, that's one of the scary things. If they do it before childbirth and then people start selecting, okay, I don't want that baby because it might have this birth defect. And we already do some of that. It's just not as sophisticated as this. And so I hope they use it as a curative method, but people always take technology, and people ask the same question when we started doing cytogenetics and we could see broken chromosomes in cells. That people ask the same question, will this be used for bad things? Or we could see 2 copies of a chromosome or 3 copies or 1 copy, and we could figure out, oh no, there's a genetic defect. What do I do with this child that's got this genetic defect? So we're looking for genetic defects. We're just looking at a much more fine perspective, and hopefully no one would ever say, okay, this kid might have cancer when he's 50 because he's got this mutation, but hopefully rather they would figure out a way to prevent that progression from happening. And if we can see that, hey, when Americans are born, 50% of us are already predisposed to cancer, hopefully that will get people even more excited to figure out ways to fix that from birth. And if we could fix it congenitally while they're still in the uterus, then that would be the easiest place to fix it, because there's a lot less cells that are involved in the human body at that point. And so I always hope for good. I'm always optimistic, but I always know that some people use information for the wrong purposes too. All right. So that's what we're talking about here when we're talking genomics, and basically we're comparing those sequences to known sequences, and sometimes we just presume the function because we don't know specifically what that protein is going to do when it's actually produced inside of the cell. And then we get to this field of metagenomics, and metagenomics is then, slide stuck, oops. So metagenomics is analyzing the entire population, like in your gut, and trying to figure out what every organism that's out there can do, how they work together, which ones need each other, which ones don't like each other, what antibiotics can they make, and all of those things. And that's another aspect of genomics and metagenomics. We know of about 400 different strains of one species that lives out in the soil, and they all make different antibiotics. Some of them are really, really good at killing other microbes. About 450 of them have been discovered. Different antibiotics have been discovered that soil microorganisms make of one particular species. It's the Streptomyces species. And they make all these different antibiotics that possibly we could use. So that's another thing that we can get out of this. What do they make that we might be able to use for something? And of those 450 antibiotics, 400 of them are so toxic that we can't use them in humans because they damage our cells too. But about 50 of them are just toxic enough to deal with bacteria, and we actually use a good number of those 50 currently as antibiotics. And so there is lots of good that can come out of this process. And so metagenomics is looking at all the genes and how all of the gene function in your gut interact with your cells. In another chapter or so, we're going to talk about bacteria actually changing their host so that their host will be nicer to them. It's not an animal model, it's a plant model, but the bacteria actually send part of their chromosome over to the plant cells, and they genetically change the plant cells so that now the plants produce chemicals that make it better for the bacteria to live in association with that plant. So we know that bacteria talk to plants genetically. They send them genetic information. They send them parts of their DNA, and they actually make the plant make molecules that help the bacteria. So are they doing that for us? We really want to know. We want to understand how they're controlling our life. Are they the ones that are saying you're hungry when maybe you're not, and telling your brain that send the signals that you need to eat more, because it's really them that are hungry, not your cells that are hungry. And so we really want to understand that. So we're looking at the microbiota, and it's important to also know what's going on at different body sites. We've talked a lot about the gut, but what's going on at the throat, what's going on in other parts of the body, and so that we can understand how disease progresses.
When we're pulling genes out of the human body, does it stay specifically to the gut, or are there other areas of the body where we can actually go in and pull out a number of different genomes and have results?
Are we talking about bacteria?
Yes.
Okay, so sure, that's what this slide is saying at that line that I just highlighted. There are bacteria all over our body. There are bacteria that are on the skin, and this is an interesting thing too, that in clinical, what's it called? In the area, criminal justice, sorry. In criminal justice, in the old days, we used to go and see if the bad guy left a fingerprint someplace when they were doing their crime. And people have figured out, hey, if you just wear gloves, you can't leave fingerprints. And so a lot of times there aren't fingerprints left at the crime. But we all have different strains of Staphylococcus epidermidis on our skin and other bacteria on our skin. Anytime you enter a room, you shed some of those bacterial. So forensic scientists and criminal justice scientists are figuring out ways to go in and sample the room for the specific bacteria that are in that room, taking them back and sequencing the genomes. And when they sequence that genome, the genome that's on the bacteria that they find is very, very likely only going to match yours. It won't match Dominica's or anybody else in the classroom. And that definitively IDs you as having been in that place, because there's no other way your bacteria could get there unless you were immediately prior intimate with the person that was there, shed some bacteria on them, and then they fortuitously fell into that environment. So each one of the staph epidermidis on our bodies have different mutations that none of the others of ours will have. and they can look at those mutations, and it gives a very nice genetic fingerprint of where that organism came from. So yeah, this is going really far, and it's going to be really powerful down the road, and hopefully none of you have to deal with your bacteria being in places that you wish they weren't down the road. Okay, so we can use it all over the place. We can study organisms that live out in the ocean. We could get lots of data, but with lots of data, there's lots of challenges. And so as the data grows, computer systems and analytical systems have to grow. And the government in the U.S. is being pretty good at continually funding those projects so that we can continue in that research. All right, that's the end.